So now let's move on to understanding how some of these organs regulate blood flow locally. This is a very important concept that we need to explore further. It is called autoregulation because whatever the organ produces within itself regulates vasodilatation and vasoconstriction. That's why it is described as how blood flows to an organ remains constant over a wide range of perfusion pressures because the organ can regulate it itself with metabolites of its own cellular respiration. There are also some external components that help regulate, particularly in organs that are dependent upon the autonomic nervous system or other neurohormonal axes for regulation. Let's start with the heart. This is the cardiology section. It is really convenient that the products of cell respiration can modulate the coronary vascular tone. So for example, if the heart is beating much faster and harder, the mitochondria within the myocytes will be working hard to make more ATP. Eventually, we run out of oxygen and there will be no oxygen to make ATP. Then we just start accumulating the precursors, ADP, AMP. Now what does the cell do when it starts accumulating a bunch of these? Well, it's not just going to let them sit there, it's going to break them down into another precursor, which is adenosine. Why in the world would we want to break it down into adenosine? Well, we just used up all of our ATP because we exceeded our supply of blood because our demand is very high. What we need to do is to increase our blood supply again. Remember, at baseline, we use 80% of the oxygen that is delivered to our coronary arteries. So we max out really fast if we increase our demand. We need to vasodilate. Can you guess what molecule on this page is going to do that? Adenosine. How convenient that the breakdown of precursors from cellular respiration will help us when we can no longer perform more cellular respiration. So adenosine has autoregulatory effects by binding to two receptors, A1 and A2. A1 receptors are on myocytes and A2 receptors are on vascular smooth muscle cells. Now, what is important here? Well, when you bind A1 receptors, you activate the G inhibitory subunit. This means we get a decrease in cyclic AMP. And we are pros with knowing what a decrease in cyclic AMP does within pacemaker cells and myocytes. Number one, you decrease the funny current in pacemaker cells, which means that we decrease our heart rate. In our myocytes, we decrease the opening of L-type calcium channels, and we decrease calcium-induced calcium release because of the decrease in L-type calcium channel opening. We also decrease intracellular calcium. This all leads to a decrease in contractility. And this is all due to low cyclic AMP causing protein kinase A to remain inhibited. Remember, no protein kinase A means that we don't activate the funny channels because they are dependent on cyclic AMP for opening. We don't open these channels because they're opened when they're phosphorylated by protein kinase A, which means that we don't have an entrance of calcium into the cell. No calcium will be available to bind the ryanodine receptor on the sarcoendoplasmic reticulum, which means that there will be no more calcium release, and we will have no substantial increase in intracellular calcium to bind the troponin C. And we do not have protein kinase A phosphorylating phospholamban, so phospholamban activity is increased. Very different in the vascular smooth muscle cell is that it is associated with GS. Remember, in vascular smooth muscle, not myocardial muscle, an increase in cyclic AMP causes protein kinase A to have a inhibitory effect because it will add a phosphate to myosin light chain kinase. Myosin light chain kinase will not be able to do what it does. It will not be able to phosphorylate the light chain of the myosin head, which means that you get no contraction of vascular smooth muscle cells. Therefore, we get a vasodilatory response that we were looking for. Excellent. Now, what about nitric oxide? we know that nitric oxide induces guanylyl cyclase. Let me write it out for you because we need to be able to spell these things because I usually forget. So guanylyl cyclase means that we increase our cyclic GMP levels, which will cause vascular smooth muscle cell relaxation. So this causes even more dilation of our coronary arteries. Now let's go on to the brain, where we can discuss carbon dioxide. So the carbon dioxide levels in the brain are very important. If carbon dioxide is high, it will combine with water, and via carbonic anhydrase we produce carbonic acid. Carbonic acid will dissociate into a proton and bicarb. High levels of protons within the brain will cause vasodilation. Why? Well, the brain is a very unique organ in that high levels of carbon dioxide will cause the arteries to open up because they want more blood to come in and deliver oxygen and then take away the carbon dioxide, because the brain always wants oxygen no matter what. It's the most important organ to deliver oxygen to. So again, how does the brain try to increase its oxygen supply? By hypercapnic induced vasodilation. This is a concept that you will see used in patients that have really high intracranial pressure, usually due to hemorrhage or swelling that is causing a mass effect. Remember, mass effect just means that in the limited space of the cranium, there is something there that should not. Whatever that is, it is compressing the structures that normally sit in our skull. One of the methods that neurosurgeons use to help reduce this increased intracranial pressure is to decrease the amount of blood being delivered to the brain. How do we do this? We hyperventilate them, so we increase the respiratory rate, which drives down our CO2, which means that we vasoconstrict. And that is how you decrease blood delivery to the brain, because you don't want more blood to leak across those damaged capillaries in that area that is necrotic or edematous. And by also vasoconstricting, you decrease some of the pressure in the brain. Remember, this is a very key concept in managing an increase in intracranial pressure. We manipulate these normal physiologic principles to take care of patients, and test question writers will make sure that you know these normal physiology concepts. Now, what about the kidneys? We discussed the kidneys in depth when we talked about the RAS system. Remember, we discussed the kidneys have stretch receptors in the afferent arteriole, so the myogenic feedback refers to the afferent arteriole because it has smooth muscle, and so does the efferent arteriole, and the tubuloglomerular feedback, so the tubules, which is the macula densa cells, sensing sodium, and then sending signals back to the JG cells to release renin to manipulate our GFR, or manipulate the hydrostatic pressures in the glomerulus. Remember, the macula densa also produces cyclooxygenase 2, which will mean an increase in PGE2 and PGI2, and this will cause vasodilatation of the afferent arteriole. This is a unique situation that you must know. And the same thing goes for the lungs. The lungs is an incredibly important concept to know. Hypoxia, or low levels of oxygen, causes vasoconstriction. What do you think the lung vessels will do if there's low oxygen tension in the alveoli due to some obstruction or fluid accumulation within them? Do you think they will waste sending precious blood flow near these alveoli that cannot oxygenate them? Remember, the purpose of the lungs is to take oxygen from the alveoli and put it into the blood. If there's low oxygen in the alveoli, you're not going to send blood there because you're not going to be able to put oxygen in it. It's wasting precious blood flow. The pulmonary vasculature will vasoconstrict in areas where oxygen tension is low. So if you have our alveoli here and the O2 is very low, here's our vessel. Instead of go here and send that blood, it's going to divert and enter this vessel and go to a different alveoli somewhere else in the lung that has a higher oxygen tension. So therefore, oxygen can flow into the vessel, and then we can take the oxygen to other parts of the body. This is really advantageous unless you have a situation like congestive heart failure. I will discuss how this plays a role in CHF when we reach the pathology section. But a quick preview is that in CHF, we have a backup of pressure and volume. P is pressure, V is volume, and this will actually cause fluid to leak from the pulmonary vasculature into the alveoli. So these alveoli get filled with fluid. So they basically get obliterated. And fluid cannot take in oxygen from your lungs. Therefore, these vessels cramp down because they vasoconstrict away from areas that have no oxygen. And then this further causes an increase in pressure because we are decreasing the diameter of these vessels, causing an increase in resistance, which is R. Now what about skeletal muscle? Muscles are always undergoing high rates of cellular respiration if you're using them. So accumulate lactate, potassium, protons, carbon dioxide, and adenosine because we break down ATP precursors, which are ADP and AMP, and we get more adenosine. Now, what do we need to know from this slide? Remember that CO2 will produce a proton and bicarb via carbonic anhydrase, and then it will dissociate into carbonic acid components of a proton and bicarb. The potassium is released from a lot of musculoskeletal contraction, and this can hyperpolarize our vascular smooth muscle cells, causing vasodilation. We discussed how adenosine stimulates the A2 receptors, causing stimulation of the GS protein subunit, which will lead to an increase in cyclic AMP, which will activate protein kinase A, which will slap on a phosphate group onto myosin light chain kinase, and therefore we get vasodilation. So now, the last one we need to discuss is skin. Skin has a strong microcapillary network that is highly regulated by the binding of alpha-1 agonists. So activation of the sympathetic nervous system will release epinephrine into the bloodstream. When it binds alpha-1 receptors, because it's an alpha-1 agonist, in the microvasculature, you will have high levels of vasoconstriction. This will actually decrease temperature dissipation because the blood vessels are far away from the skin surface. If this is our skin surface, we want our vessels really large and close to the skin surface so heat can dissipate. But at the same time, when you have alpha-1 agonist activity, you want blood to go to more important places. Remember, when you have the sympathetic nervous system activated, it is our flight or fight response. We want blood to go to the brain, the muscles, and the heart. Excellent. Did a really good job there, guys. Keep up the great work. Alright guys, this is our last flash quiz for this session. By what mechanism does increasing the size of the heart cause an increase in myocardial oxygen demand? So in what way did we increase the size of the heart, and by what mechanism did it lead to an increase in oxygen demand? So we're experts on this. This is an increase in wall tension. Remember, the law of Laplace states that wall tension is equal to the pressure within the ventricular chamber times the radius divided by wall thickness. Anytime we increase the pressure, the radius, or if we decrease the wall thickness, that will cause an increase in wall tension. Whenever you cause an increase in wall tension, you cause an increase in myocardial oxygen demand, because wall tension is a force that myocardium will have to oppose directly. Remember, if we draw our ventricle like this structure, this is our radius, the pressure within the ventricle, this is our wall thickness, and then this is the wall tension. So if you distend this chamber with a bunch of fluid or a very high pressure, you're going to increase the wall tension because you're going to increase the pressure, you're going to increase the radius. Our myocardium will compensate to very high pressure by increasing the wall thickness with concentric hypertrophy. If we add sarcomeres in parallel, which we do with concentric hypertrophy, there is more myocardium. It has to work harder. It also gets less blood into the ventricular chamber, setting us up for a problem because we have increased oxygen requirements because we have more muscle, and at the same time we have less fluid in the chamber to forward perfuse into the aorta and supply the coronary arteries that sit at the root of the aortic valves on the distal end. And in the situation where we have increased volume, we increase the radius, and this adds sarcomeres in series, which also increases the wall thickness, and it allows our chamber to really distend a lot more because it's a volume overload problem. And this is the situation of eccentric hypertrophy. In both situations, we increase the wall thickness, which means we have more sarcomeres, more myocardium, you require more oxygen. These are compensatory mechanisms to that initial increase in wall tension that will eventually lead to an increase in oxygen requirements.